It is not just organic food alone, but we must precisely know what we are eating and how much bacterial load it's carrying. I'm not a clinician, so I'm a geneticist. So what I'm saying is genes are not immune from nutritional effects. The soil is slowly becoming unsuitable for agriculture. Now, if genetic engineering is doing the job of a god, then I pity God. Plant biomatter on the planet of Earth is about 45,000 crore tons, and we are zero point zero 0.06 crore tons. So, this is a holistic approach
Wow.
against the pharmaceutical approach, and that is the absolute biological reductionism. For example, if some people eat a lot of food, still they are lean, and some people are don't eat that much, but they are obese.
Woof woof. Welcome to GhanaCast.
Today I'm speaking to Professor Arjula Reddy. He is a biologist who has specialized in the study of genetics, genomics, and biotechnology. He is a fellow of the Indian Academy of Sciences, and he has collaborated with the Max Planck Institute in Germany, Purdue University, and the Rockefeller Foundation.
Namaste, Professor
Namaste.
Arjula Reddy. It's such an honor that you are here in Kana Shantivanam, and it's such a privilege that you took the time out to record this interaction with us. And you, of course, are a pioneer in the field of genetics in India, and also you have been involved with education. You've been Vice Chancellor of 2 major universities, also on the board of governors of the Banaras Hindu University. And you've seen the field of genetics grow. I mean, you've nurtured it in India, and you must have seen a lot of change happening, how life has changed, how our lifestyle has changed. So currently you are involved in a very interesting initiative about that deals with the problems that come with these changing times. Can you tell us a little bit about that?
Yeah. Yes. Namaste. It was I'm really fortunate to be here in Kana Shantivanam and share my thoughts. Thanks for inviting me. Now, last few decades, environment is changing rapidly. Climatic aberrations can be seen. Frequencies of natural disasters are increasing, and human health is going through a very rough patch. So I thought, well, we have science to answer some of these, but science alone cannot do this. We have a role to play in different manner. That is, the science of health originally and the concept of health. With so much pollution around, so much degradation of soil, so much degradation and so much harm, damage to the plants and plant produce, which will have direct effect on human systems, prompted us to start a concept called One Health. One Health basically means is the health in a series that is fundamentally starts with the soil health, plant health, and human health. The common factor in all these is the soil microbiome or soil microbiota, and then plant microbiota, and then the human gut microbiota. If we concentrate only to protect the health of the humans through medical interventions, we are totally ignoring what happened to the other two sources where the problems might originate from. So genetically speaking, all these three systems, the soil microbiome in the rhizosphere, and plants systems, and a human, all share some of the common microbial fauna. In other words, the soil contains several genera of bacterial species, several genera of bacteria. And plants also contain, so is the human intestine or gut and gut epithelial cells. What is common is the soil microbial diversity is similar to the human gut microbial diversity.
Wow, that's amazing. I mean,
Yes, and then their ambience, their inner atmosphere in which they live, so-called we call internal pH, is almost similar.
mm,
So it is soil microbial systems some way influence the gut microbia. But how the soil microbia get to the human gut? Very simple, through the plant foods or animal food, but mostly plant foods. So when you take just apple fruit, you're taking in a lot of microbial cells and bacterial species and other microbes. So if you want to improve the human health, we must understand what is the beneficial bacterial proportion in the soil to the total bacterial population. How much of that is beneficial bacteria, how much is not beneficial bacteria. Similarly, in the animal, in the human system, I'm not even talking animal, that's a different world again.
Yes, yes.
But here the context is human health. We have been eating indiscriminately. So therefore, the human, the soil microbia indirectly affects the balance of the gut microbia. And then in the process, the bad bacterial composition will increase in the human gut, and that's the problem, leading to gastrointestinal problems. And what is now coming up is this gut microbial health or gut microbial balance, equilibrium, hurts. If it is hurt, it hurts not only the gastrointestinal ailments, but it is now linked with several other diseases that are known to the mankind for a long time, like Alzheimer's, hypertension, diabetes. So these are slowly coming up. For example, if some people eat a lot of food, still they are lean, and some people are don't eat that much, but they are obese.
Correct.
The problem is how much of the food input is absorbed, how much is digested. That is uptake and digestion and out. Any of these processes will decide this. So these bacteria have a role in that. So as a geneticist, when I thought, all right, we will go for a metagenome sequencing, identify what is the proportion of beneficial bacteria and non-beneficial bacteria in the soil around a particular food plant, whatever is supplying the food.
Yes.
And then also see whatever is the composition of the human intestinal bacterial microbiome, and compare the genomes, and then easily identify the equilibrium or the otherwise. So in this process, we don't look at just bacterial diversity alone, that is, which bacterium is of more. For example, bifidobacteriums are more in some cases, then lactobacillus is predominant in other cases. We are not only looking at that, we will not only look at that, but we will also look at epigenomic effect. So how these bacteria are playing a major role in vis-a-vis the epigenome effect. So epigenome sequencing is also essential in determining this. Net result is we have to design a precision diet based on data of gut microbiome and a patient's regular intake of the microbial fauna. So you require to sequence the fecal source and also the epithelial swab or epithelial scrape samples. It requires a lot of metagenomic sequence and epigenome sequencing. Ultimately, we think that some of these nutrients in the plants which are depleted because of this artificial heavy dose of fertilizers, heavy dose of pesticides, herbicides and so on, are creating a situation where epigenomic changes are occurring rapidly. This is a theory. it will come out if we have a large sample of analysis. Now the system has just begun.
Correct.
So people are now thinking that it is not just organic food alone, but we must precisely know what we are eating and how much bacterial load it's carrying. Organics doesn't mean divide of bacteria. No. In fact, organic food may contain more bacteria because we are not using antibacterials or anything there, chemicals. But they may be beneficial bacteria.
So we need to know if they're good bacteria.
We need to know that. So therefore, while defining a precision diet or a curated diet based on the genetic and epigenomic data, then we have a claim to say, yes, if you take this diet, you are getting these, and then we prove through epigenomic results. So this is the concept. So One Health, you cannot separate human from plant, plant from soils. It's all linked. It's all linked. So this is a holistic approach against the pharmaceutical approach, and that is the absolute biological reductionism. So you look at a part by part.
You look at the gut and you treat the gut.
You treat that only.
Yes.
And okay, you have a problem in the throat and you only treat the throat. But that came from sinus or someplace. So these are the problem. But I'm not a clinician, so I'm a geneticist. So what I'm saying is genes are not immune from nutritional effects. They're not immune. And nervous system is not immune for such changes through aberrations in the intestinal atmosphere. So how to delineate them? This only requires sequencing and identifying the interactions between gut microbia and plant food-based microbia, which is essentially carried from the soil microbia.
So in the initial studies, have there been sequencing of soil samples done to check the microbiome? And what have the initial findings been? Is it deteriorating over time? Is the bad bacteria increasing, the good bacteria decreasing?
The soils are eroded with chemicals and runoff water, repeated tilling, deep tilling, surface topsoil that we call in agriculture, that is getting eroded. And in some places in Punjab, Haryana, and in other places, very few places, the soil is slowly becoming unsuitable for agriculture.
Wow.
It will happen in another maybe 10-20 years. That is because continuous erosion of soils. Now, in that case, to nurture the soil, you need to intervene biologically. And this government is now laying emphasis to go for biologicals for every agri-chemical that's possible. It's a national strategy. For that to happen, a lot of innovations, a lot of molecular genetics and genomics, and bacterial engineering. So these will happen in future, no doubt. But right now, the system, we are at the stage of only identifying the problems. And it's still not established, really in real genetics, it is still not established whether it is the diseases that cause imbalance in bacteria in the gut or imbalance in the bacteria that cause the disease. These two are different things and require different advanced techniques to delineate this problem completely at the genetic level, at the chromosomal, at the gene level. So that has to be settled, and it will be sooner or later. But in a country like India, where we have huge biodiversity of plants, microbiome, and even human genetic diversity is very high.
Absolutely.
We should now, we cannot ignore soil microbia and plant microbial species as a resource. We cannot ignore.
Absolutely.
We cannot ignore. And plant, for example, plants constitute plant biomatter on the planet of Earth is about 45,000 crore tons. And we are 0.06 crore tons, humans.
So it's just a fraction.
We are nowhere in the picture, but we are destroying that.
Yes.
That has to be understood.
Yes.
And we can destroy the plants only up to certain level. After that, we are destroying ourselves. That's what it means, basically. Oxygen depletion will occur and so many other things will occur.
We are so dependent on
Yeah,
plants.
because of this distortion, the climate change and all that, the frequencies of natural disasters all over the world, like floods, like forest fires, increasing tremendously. The frequency is increased very high. And hungry map of the world shows two types of hunger, two types of malnutrition. One is undernutrition, one is overnutrition. In both cases, nutrient supply through the food is not optimal.
Not
because
adequate.
of the soils gone. So under these circumstances, we have to see that we have to respect the overall biodiversity on planet Earth. I will give you one example. There are small worms we call nematodes. They are around the root zones of the crop plants. Nematodes, they are called worms. Small worms, where huge genetic work has done on them, inside out. Now, some chemical companies invented nematicides that will kill nematodes because they eat roots. Now, what they didn't realize was it is the nematodes, they eat bad bacteria around the soil. Now, the chain is disturbed. So, the bacteria population goes up. So, instead of nematodes eating the plant roots, the bacteria started doing this.
Oh.
So, it's a very complex chain.
It's like a web that you...
It is a complex web. And we are disturbing repeatedly. So, what I call sometimes, we are at a point of crossing the planetary borders. In biodiversity, we have already crossed the planetary boundaries. That is Earth. You can't come back. Such as disappearance of species. We cannot come back. They're gone.
True.
We can clone some, but not all. Millions of microbes disappeared. So, now next one is temperature differentials. That's going to happen. Third one is water. And the last one is, of course, oxygen. So, if we keep on doing this, if we just take care of only human health, that's not enough. The root problem lies at the base, the soil. That is the one which nurtures human life with water and oxygen. So, therefore, one is linked with the other. So, the eating, as the old saying goes, you are what you eat.
Yes.
So eating at the same time, the community level, foundation level, well-meaning people, well-wishers of Earth should look for a holistic problem rather than a isolated problem of just human-centric, anthropocentric issue. This is not an anthropocentric issue.
This is so fascinating. Actually, a lot of people feel that they can buy vegetables and fruits and then they dip them in potassium permanganate. But what you are saying is that the microbiome is actually carried in the plants, you know. Yes. So the bacteria is being carried in the plants. So you can't really wash it off with...
Yeah. If you start washing with vinegar or with 2% acetic acid or something like that, or even this potassium, sodium bicarbonate and all that, there is a likelihood of some bacteria getting washed away. But if you go out of your home, go to the open space anywhere, and just fistful, just close the fist and come back to the lab and check what is there in the air. You see substantial percent, up to 40% fungal spores in it. So these bacterial species spores, fungal spores are freely there in the air, and they are more in a polluted air as a particulate form. So there will be a lot of chit chat between these genetic apparatus of one bacterium with the other one. Actually, when two bacteria interacting, obviously through their gene products, and ultimately it's all in the genes. So you will see that such interactions you cannot ignore them. So we have to see, if you have a healthy crop around, if you have a healthy air around, that percentage will differ. dwindle actually, will get low. So we can understand in that sense. But here what we need is advanced technologies, for genomic sequencing, whole hog genomic sequencing for human and also plants and soil microbia, metagenomic sequencing and epigenomic sequencing, and collating this data to the clinical analysis of these patients who are suffering with gastrointestinal diseases or gut microbia linked diseases, indications. So that will give some idea of what to eat, what not to eat, and how to eat. If you just cut the vegetables from the plant when you buy from the shop, and then wash them and cook them, you all those nutrients, you do not know how many of those nutrients, except micronutrients, how many of those nutraceuticals retain their integrity. So the green salads we have to eat, but green salads you eat, that means more bacteria will come, both
good and bad.
But for some time, if you keep on eating, you will adjust to it. You can tolerate bad bacteria. It's better than eating chemicals.
Yes.
Okay. So immune system will rebounds. We have innate immunity for this. So this is the way we, the genetics people, think that ultimately we have to look at the DNA level to see what's happening. Why some people don't care what kind of food they are eating, how much bacterial load they can take. They don't care. Nothing happens to them. So there is something. And that is in their genes only, nowhere else. So it's either epigenome or normal genome. So we are now at the crux of doing the whole genome sequencing, targeted gene sequencing, metagenome sequencing, and then correlate all this data with the physical data, with biochemical data of the nutrients of the what are the nutraceuticals that are present in a given vegetable, in a given season, given under the type of growth conditions, cultivation. Then correlate it with
the clinical.
Yeah, because in the human history, as on today, in the entire human history, nowhere, never humans exercised food choice as on today.
Yes.
They're not only asking for their choice, that no, not this food, not that one, not this millet, that millet, not proso millet, finger millet, not finger millet, foxtail millet, no, pearl millet. So all this is a revolution in the food choice industry. Now, food choice area. And not only that, we are reading now in the back of the packages how much protein, how much saturated fatty acid, unsaturated fatty acids. All this we are reading. This is going to propel a revolution sooner or later on designing foods. That's when these precision diets will come.
And that genome sequencing, of course,
yes, will be backed by not necessarily whole genome sequence. We have targeted genes. For example, there are some genes which are known to play mischievous role in protease, in stability of the protease, or the other way, glycosylases, hydroxylases, some of these phosphatases. So these are the target enzymes. We can look at those targets. And the bacterial villains, we know what are those targets, so we can also look at them. That's a little easier than whole genome data collation and gene mining, and then targeting.
Because
clear.
It's a huge effort,
Yes,
sequencing is a huge effort, and more than that, the data analysis after
sequencing is a little bit more easier effort than analysis.
Yes,
that takes
huge amounts of
that
data.
takes long time. And now with artificial intelligence coming in, probably we will get better off.
Yes, because it can recognize patterns.
Yes. So there are companies in Hyderabad, everywhere, like one of these companies which is propagating the idea of precision diet is a Yellow Life. Their concepts are very clear along the lines that I just said it.
Yes.
There may be others, but it's not an easy task to make a name in this field, because there are lots of unknown things.
It's a brand new sort of
New
field.
field. So there will be first comer advantage. There will be first comer disadvantage also.
Absolutely.
But as far as science is concerned, I am very happy. I have been from the beginning since last almost 50 years after my PhD, thinking of integrated biology and holistic biology, not reductionism. Typical biological reductionism leads to molecule-centric treatments, molecule-centric approach to cell-specific diseases, organ-specific diseases. But the problem is with the whole body, not just genetics is the answer, and this is slightly is now slowly coming up. That is really a fortunate situation, a happy situation for me.
Absolutely. A lot of traditionalists say that, you're talking about precision diets. A lot of traditionalists would say that we had already achieved that in a way in the small areas, the local diets that communities had 100 years ago, 150 years ago. But do you think those are, now we are at a point of no return, that the soil is so deteriorated everywhere?
No, two things here. First thing is, we are giving better food, we think, that is, a little better clean and neat and all that. That's step one only. Step one is we are hygienic food, carefully selected food and all that. But that's not based on the data. That's based on our perception, based on the field conditions, probably. But what now new things coming up is we can show the basis of that. Why you are saying that this fermented bread is better than refined bread? Okay, normal bread. Okay, fermented vegetable we can say we can keep it 6 months, just like that.
Absolutely,
no problem. So that is what I am saying. So one way, step one is that good diet. Step two is the precision diet. That takes time. That's what I am saying.
Absolutely.
And right now it may be expensive because so much data is going into it for precision diets and all that. But sooner or later it's not going to be that expensive.
But I think the main challenge perhaps would be, of course, to spread the awareness of this,
yes.
that the soil is so important. Because coming from a small village myself, I know that the farmer is just looking at whatever
yield,
whatever,
survival.
If there is a pest, he doesn't care of what he's putting, as long as it gets rid of the pest.
Yes.
But that correlation, that this going into the soil is then going into your stomach and is then creating problems in your gut, that awareness is an issue.
Yes. So this awareness of the farmer, no matter how much you tell, finally his survival instinct.
Absolutely.
So survival, to make it farming profitable, he has to do all this. Now, what we can do, science people can do, is while awareness programs go on by the government aided agencies are there, some NGOs or the scientists are, what we have to tell them is give them technologies that will replace or substitute gradually synthetic chemicals that are used to kill the insects, to kill the pests, to kill the bacteria and all that in the fields. Right? And that is also causing environmental damage and even human health, because it is accumulating, concentrating. Some of these herbicides and some of these pesticides are concentrating in the young mother's breast milk. It's known. So we have to evolve methods of organic crop protection. For that, we have to use biologicals, not chemicals, like pesticides. So there are now, this is all linked.
Absolutely.
In the integrated pest management system, we slowly replace the synthetic chemicals like herbicides and pesticides by naturally produced, environmentally safe, benign pesticides or benign molecules that will do the job in a different way. That requires innovation, and that is done also. That's slowly it's done, but it's taking a long time. No technology, no new technology will spread to the field so easily.
True.
It requires nurturing by the government, policymakers, and progressive farmers, farmer cooperatives, and NGOs. Otherwise, it's very difficult to run.
And
even if today we have a product in Hyderabad, we have a product that replaces substitutes to a large extent a pesticide, very well known, government supported. But to spread into 1 lakh acres, it took us almost 8 years now.
Wow.
So it will now take off stage, but there is no awareness. Now there is awareness.
So very slow.
Very slow, but now it will catch up. See, when BT technology came up, it was resistance earlier, but it spread like no other technology ever in the human history. In India, after it started, within 3 years, it spread to 10.2 million hectares. That's a different story. But in any case, what I'm saying is, all revolutionary technologies are not standalone technologies. They have to be supported by policy interventions.
Absolutely.
And also nurtured by the government and their agencies.
Absolutely, because you have to be competitive against established practices.
Yes. I didn't want to broach into that because it's sensitive. It becomes sensitive.
Yes.
In a government like this, in democracy like this, we have different interests. We have different competing interests.
Yes.
So we have problems.
Yes. But it's a very, very fantastic area of work that One Health is doing. I mean, making the connection between soil. This is probably part of the thing that will address the problem, because the awareness will spread.
Yeah.
I mean, apart from the actual genome sequencing, just the awareness of the work of One Health will help more people know about how the soil directly affects your health.
Yes.
Absolutely. So, sir, you have been involved in the genetics field right from the very start.
Yes.
And how did you make this choice of genetics?
Yeah, I graduated I did my BSc Special Chemistry. No plant background at all, undergraduation. But when I was working on a small research project in chemistry at Osmania University, I realized that the plants, these chemicals, that the problem given to me is produced by plants. And those are very good antioxidants. Now, all pharmaceutical industries now, they are selling their antioxidants.
Absolutely.
But the plant has full of them. So then when we worked and realized, okay, there is an antioxidant. But why antioxidant is important? Because it has a lot of hydroxyl molecules hanging around. So they can easily trap the free hydroxy radicals and so on. So it will anti-inflammatory and all that. So then I thought, okay, then obviously there must be a homeostasis, the mechanism by which neither too much of free oxygen radicals exist or free radicals exist, nor too low. That means there must be a genetic system balancing this.
Controlling.
And plant is producing it, and we are eating the plant products. There is no other way. I mean, animal food, okay, once in a while, every week, once maybe non-veg, but basically plant eating.
Vegetarian.
So there I got interested, okay, in the food grain plants, so what are the antioxidants? So the PhD was in maize. So I was looking for genetics. So they were not giving me seat in genetics because I have no background there. In BSc, it is all chemistry, so special chemistry. So I fought for it and got it. Only 2 departments of genetics at that time, bona fide genetics. One is Banaras Hindu University, Department of Genetics. Professor Rai Chowdhury was the stalwart in that. And Osmania started. He started Osmania Genetics Department.
Wow.
He advised the government and started here. There is one in the Department of Zoology. There are several units of genetics in Department of Zoology is botanist, but Osmania University had a genetics. So then that fascinated me and I got in. And then I worked for PhD in maize and then went abroad and then ended up in Johns Hopkins University Medical School with a very peculiar problem of moving an insect gene into a plant gene into insect gene.
Wow, that must be fascinating.
In those days, 1976, and the paper appeared there. Very tough. So then I came back to Hyderabad University and never looked back. And then rice. Most of the work is on rice now.
Wow. But after Johns Hopkins, you must have, the world would be open to you because such a prestigious institute.
But I wanted
What prompted you to come back?
This is my hometown, number one. And the vice chancellor there, Gurubaksh Singh, he talked to me on phone in those days. Ajao. So Ajao means no appointment letter, nothing, no interview, nothing. But I came, trusted him. Ha gaya, jao. Ho gaya. That's it. And we have joined somebody from Harvard, one fellow from Chicago University, and I am from Johns Hopkins. 6 of us in one go joined. Today, that Hyderabad University in such a short time started in 74, and today it is an institution of eminence.
Absolutely.
So its foundation was like that. Then I started department. My colleague also, there was a geneticist. So we started and we grew full. But there is nothing like independent departments at that time in Hyderabad University. There are schools of life sciences, School of Life Sciences. And that genetics division is there.
Okay.
Genetic group.
So there must have been many challenges also. Such a new field.
There was a shed. We were working in a shed, stools and no chairs, only stool. All that. So, but determination was there. Do something which we can show from India we can do.
Yes,
and that's what we have done.
Getting the university and all the other universities to treat this subject seriously.
Once we established Hyderabad University, then papers started rolling, and then we got tremendous respect. We were in every committee. So then we knew how to promote genetics in other places.
Absolutely.
Yeah.
And then you went on to also mentor a lot of people.
25 PhDs.
25 PhDs. Wow.
Some of them are leading companies holders now on this on genetics, in genetics. Some of them are good academicians, and 1 or 2 of them are in pharmaceuticals. But genetics is their forte. Because I talk all the time in the lab on holistic thing. So some of them are doing holistic work in that chain of events.
Wow.
In that chain from soil, plant to human. A very powerful group of students are working on the plant itself. How to stop insect eating away the plant. Season by season.
So how did you develop this holistic approach? I mean, there are very many geneticists just studying a particular...
insect, particular organism and all that.
Absolutely.
Yeah. Because my training was, I worked on maize all the time. Corn, maize is corn.
Yes. And
then insect, Drosophila insect. It's famous for genetics at Johns Hopkins. And then here I come maize rice. So it is a overall. Okay. Now, then coming human. For a geneticist, it doesn't matter which organism. See, when we whole genome sequencing is done for humans, it came out. The human genome is not the autobiography of the humans. It is an autobiography of all the animals below the evolutionary line. That means we have a little bit of donkey's gene, little bit of monkey's gene, little bit of dog's gene, little bit of this, little bit of that. And we are 98.9% or whatever that is similar to chimpanzee.
Oh, so you're just seeing the tip of a big tree.
Yeah, we are so close to chimpanzee. Even gorillas are not that close to chimpanzee.
Wow.
Yes.
So we are closer to chimpanzees.
Yes.
Than
the reason is, but then you can ask me why, if the 90% genetic similarity is 99 point something genetic similarity is there between chimpanzee and why don't we look like chimpanzees? Okay. So the point here is that remaining mutations, if you calculate it, at least there will be 3 million different changes in the genes of these. And they are all in a crucial genes. They are all in crucial parts of the genome.
Oh, okay.
So that makes the difference. There are crucial genes, very critical genes. They are the changes. For example, if we are 99.8%, then why chimpanzees don't talk like us? See, this question was asked to Sidney Brenner, Professor Sidney Brenner, who worked on a nematode, mapped every cell. There is nothing unknown in that, inside out. And when this question was asked to him, he said in a lighter manner, yes, don't underestimate chimpanzees. They know how to talk, but they also know if they talk more, they will be in more troubles. So it all went down to a bunch of genes where the muscles that control the vocal cords are the genes controlling those, genes encoding proteins for those are mutated so many times. So the mutilated gene is present in chimpanzees.
Ah, okay.
So our gene is different from that. So many mutations are there. So like that, there are so many mutations. So many mutations.
So I mean,
So
even a small difference contains this many
it
mutations.
depends where in that.
Where it,
Where that different. Suppose if there is a gene that's a control point for your behavior, let's say, a behavioral genetic apparatus, there are crucial genes, some of them, or a vision gene. A crucial gene, a mutation occurred there, it will have impact. But 100 mutations occurred in a non-important gene, in non-coding area, not damn thing, who cares, nothing happens. There is a buffer in the human genome. Hundreds, thousands of mutations can occur in those regions, nothing will happen.
Wow.
No direct impact.
Wow. One of the things that is talked about a lot in genetics is the transposons,
Yes,
And
that's my field of work. The PhD is on that.
oh, wow.
Yes.
Wow. And a lot of people, a lot of people say that this is evidence of intelligence at a genetic level, because it's like by design there is this copy paste happening or cut paste happening of the transposon. Have we understood enough about transposons yet?
Yeah. Well, I can go on, but let me tell you, there are two types of transposons. One of those two is a DNA transposons. Those are limited in number. They are found first in maize. There are RNA-based transposons, retrotransposons.
Correct.
Those are basically integrated retroviral sequences. So those are the so-called retrotransposons. So they are in RNA form, they will become into DNA, then they insert into chromosomes and they do all kinds of things. Okay. Now, again in that, there is one is a cut-paste mechanism, another is a copy and paste mechanism. These elements, some of these elements, those are autonomous. They have at their end of these elements, that's 5' end and 3' end, the beginning and the end, they have unique palindromic sequences, That's a signal that, and they encode, inside there is a gene that encodes an enzyme, very powerful enzyme, that works to excise a gene from the chromosome to insert the same thing into other place. That's called transposase. So this transposase is encoded by a transposon. So that auto-cuts it. When it is auto-cut, this will go into either on the same chromosome or into a different chromosome.
Different chromosome, okay.
Now, in the cut-paste mechanism, it is cut and the chromosome originally is relieved of it.
Yes, yes.
No doubt.
It no longer exists in that one.
But it will go into some other chromosome.
Yes.
That is cut and paste somewhere else. But copy and paste is, it copies. It's a selfish element. It copies.
Replicates itself.
Leaves the copy. And another copy goes somewhere else.
Yes.
That copy can go in the same way, 5', 3', or it will go reverse. That depends upon, if it sits in a vital gene, that gene is switched off. See, it's like a lorry sitting on an auto. It can't move.
Yeah.
Right? Now, when this cut-paste thing, when it is leaving, it will leave 3 nucleotides on both sides, replications. Leave it there. Those are called transposon footprints. We can now estimate in evolution how many times this fellow jumped around.
Yeah, how many times?
We can know that. Okay. We can also trap them. There is a transposon traps. I myself developed some, but there are others who developed a lot. That's a whole different story. It's advanced stories. But all I'm saying is, now the question is, why are they there? What are they to do with? Now, transposons also create epigenetic effects. That we know. Epigenomic effects. They create sites for methylation of the DNA to silence the DNA. Now, if an epigenomic effect, that's methylation, occurs in a promoter, the gene adjacent to it is switched off. But it's like this.
Yeah.
It's going like this direction. If it sits somewhere here, it won't move. Done. So gene, actually normal. Gene is, nothing happened to gene, but it won't be active. Now there are so many human diseases that are because of mutations. Most of these mutations, if not a large number of them, are because of this transposon jump-ins and jump-outs. And there are some disarmed elements. that sits there, it's disarmed, it cannot move.
Yeah.
No element can mobilize it.
So
Other element can.
cannot express
So
itself.
genome expansion in plants. Plant genomes are the largest genomes size-wise. Okay, 150 gigabases.
Oh, the plant genomes are larger than even
Than humans and animals.
animals.
There is no animal on earth which is as large genome as a plant called Japonica.
Wow.
Panicum japonicum. So that one has 150 gigabases.
Wow.
Human is only 3 billion nucleotides. 3 billion.
Wow.
That is 50 times more. Okay. Why such a stupid plant should have so much DNA? Most of it is this, transposons.
Okay.
Now, in maize, 75% of the maize genome is made up of
Transposons.
Yes.
Oh, okay.
75%. Out of that 75%, 85% is retrotransposons.
Oh.
Barley, 85% of barley genome is transposons. So now you can ask, human it is 30%.
30% is transposons.
So you can ask me question, how much maize is maize? How human is human? 8% of us, our genome is viral genome, typical viral genome, all kinds of viruses, bacterial genomes, and now this. So these are evolutionarily fixed. They are responsible for genome size increase and contraction also.
And decrease.
And contraction.
Yeah.
Plants, particularly plants. Plant genome size increased because of chromosomal doubling. That happens. Other than doubling, these are the transposon multiplication, proliferation that is responsible for the genome increase size.
This is so interesting.
And contraction also. You can ask me a question, if they keep on increasing,
can they contract as well?
What stopped them? Now every day it should go up. No, because there is a contraction mechanisms. Transposons themselves create contraction mechanisms.
So have we understood why this transposition happens? Why the transposons?
Okay, transposition occur under different conditions. One is, although human plant genome has it, it's silent. You give a genomic shock, such as you cross with some other species which is not compatible, genome imbalance will come. That's one. Sometimes excess heat, excess cold, high altitude. You take a crop that was growing for hundreds of years at the plains, then take it to somewhere in the middle of Himalayas with, let's say, 5,000, 10,000 feet high. Then the genomic shocks will, then it burst, transposons will burst.
Sudden burst of transposons.
Or radiation.
Radiation also causes them.
Yes.
Amazing.
We can induce transposon. You cannot create these mechanisms don't create a new transposon. They will make transposon activity more. In the process, they replicate.
You've been speaking, Professor Saab, about the epigenome, which is bigger than the genome.
Right now, it appears to be.
Appears to be bigger than the genome. So, up till a little time back, we thought that the genome expression controls everything.
Yes.
So, now are we modifying that to, because the epigenome then interferes in between?
Yeah, let me tell you, a genome controls what we are. That requires a qualifying statement. That is, if I say, I have a gene for a given disease, but the average, in average manner, to say, the gene propensity for a disease is only 30%. Rest 70% is intrinsic and external environmental effects.
Okay.
Just because I have a type 1 diabetes, I don't become diabetes. If I have a good food and precision diet and good medication and all that, I will be all right. So the question is, genome is not a blueprint of what comes out of it. No, it's like if you see, you have a blueprint of a building. You have to build according to that. Human, no, because 70% of it you still don't. What controls it, how it does. Just because you have a gene doesn't matter. You cannot dub it. Okay, this fellow has a colorblind gene and he will be colorblind. Not necessarily. It depends upon the expression of the gene, what is the proportion, what is the level of expression.
Absolutely.
Any deletions are
Because at one time in genetic history, they did think that
that is
way.
because
Knowledge was not there, I suppose.
sequencing is not clear. No, so for example, for many traits, there are more than one gene. So even if one gene is screwed up, the other one will fully replace the function, although diluted.
Yes. Yes.
So it appears that we can say, for example, the epigenome is dynamic because the changes occurring. No, it just methylation on and off, some isolation, some kind of a imprinting, all kinds of things are going on. So therefore, it is not necessarily bigger, but more dynamic. So one copy is at one stage, another copy is at another stage. No, so sequencing becomes a little tough.
So it's more responsive also
Yes,
to the environment.
Looks like it is more dynamic than the genome.
So it's
and subject to environmental variations quite a bit.
okay.
Transposons definitely create a lot of epigenomic changes. We know that.
So it's, I mean, it's not the old theory that you are governed exactly by your
No.
blueprint. Oh,
Human genome is not the blueprint of the human being, strictly speaking. There are a lot of factors that play realizing the full potential of each gene. There is a lot of factors.
That's so fascinating. So it's like, I mean, there's still a lot to study about.
The fundamental, yes, the genome is providing you fundamental basis.
Correct.
But fine print, not so strictly as atomic plant, you have strictly follow the blueprint. Here it's not so.
I remember I think this was in the 60s or 70s, where they thought that in some males there was the XYY chromosome, not just XY, but XYY.
Super male, super female.
And they thought that those men would be more violent, more prone to crime. But when you factor in stuff like the epigenome, it doesn't necessarily hold up.
No. Here, Y genome sequencing is done now. Y chromosome sequencing is done now, repeatedly done. There's not so much startling evidence is coming from there, as far as I know. But that's a whole different lot now to associate a human condition, a human indication to a portion of a chromosome or a gene is very difficult. Genetic analysis is much more complicated than +1 -1 kind of thing.
Yes. Yes, so you can't exactly
no, it's very difficult.
Say this A leads to B.
We have to do so many correlations.
Absolutely, absolutely. So there's also a lot of people, Professor Saab, who think that genetic study and genetic engineering is almost like we are doing God's work in a way, and we are interfering at a very base level with organisms, with plants, with humans also. And there was, of course, that doctor in China who tried to... So,
okay. Let me tell, because I happen to be, now that is another subject in which I am officially involved. So I have been a co-chairman for the Genetic Engineering Appraisal Committee of the Government of India. So every genetically engineered product will have to go through our committee.
Because there's so much...
my committee. I was crucial there. So this is long arguments. Now, if genetic engineering is doing the job of a god, then I pity God. Because if he can easily do his job, you can't do his job, that's the problem. No? So what I'm saying is, you are engineering one gene out of millions and billions of genes into another organism. And that gene is not a newly created. It is there in another organism already. It came, for example, a soil bacterial gene called Bacillus thuringiensis, that BT gene is transferred into cotton. Now, the insect, a specific insect, Helicoverpa, if that eats this leaf or boll, it will die. This is the concept. Now, the God party, whatever Green Party's people are, they are saying this is nonsense, this is genetic engineering, engineering. This is not the engineering of a new gene. The new gene is not created. It is gene existing in the soil bacterium, which is present in the cotton root zone also. And that one gene out of many genes is taken off and put it back into the plant. It produces a protein. That protein half-life is not more than a minute or so. So it won't damage. But nobody will believe this. Some people say by putting a bacterial gene, but the way of putting it, you have to put through a bacterium.
Mm-hmm.
How will it reach to the plant?
Correct.
So you have to have a media, either a gene gun or this. So either is protested by people because you are putting a bacteria into the plant. Plant is full of bacteria. Where is the question of putting plants in the bacteria? The typical apple skin contains more than 60 species of different bacteria. So argument is, that's a different tones. I don't want to go into the details, but that is, the argument is more against foreign monopoly on the seed industry. That is reflected in God's came to their grace, rescue. That's all I could say.
So actually the argument is against the...
Yeah, it is the foreign monopoly of the seed industry, Indian seed industry.
Very interesting.
That's undertone. And I have to fight for it. Yeah.
So, but it's also, science fiction also plays a big part in promoting this sort of fear of genetic...
Anything unknown is God. So anything unknown is God.
Yes.
Ascribed to, appropriated to God.
Yes. But since you are on the committee that all genetic
I was.
projects, you were on that committee. How crucial is control in the genetic industry? Because can there be like damage done that can be irreparable if there is not enough oversight?
No. Indian regulatory system of biologically engineered materials is very, very, very strict, elaborate. It takes a hell of a lot of time and a lot of money to get through the Indian regulatory system. Sometimes it is redundant. Several sections are redundant, used to be. Because India put in one of the Asia's or third world's first regulatory system for genetic engineering in place, so-called rules, the rules 1986, when biotechnology is nascent. That time itself we had a system in place. So our system is, if not, it is more elaborate than anything else, and it did everything to slow down our biotech industry. So a regulatory system has to be robust, ruthless.
What is the danger if you don't have a robust system?
Right. If you don't have a robust system, and if you have a loose rules and regulations and now a trial system, then long range damage will occur to agriculture. That's for sure, because you are claiming wrong things, and there is a problem. There is a problem on that, because you are dealing with artificially created, genetically created new plants, new microbia. For example, hepatitis B, genetically engineered hepatitis B. Imagine if the regulatory system is not correct, if the batch consistency is not checked, if the sequence integrity is not really checked, approved, it will be extremely dangerous. That virus will, unimaginable danger. So that is why this has to be robust.
Absolutely. On a personal, it's time? Yeah. So we'll just lastly, I wanted to ask you, on a personal level, it's very time consuming, the work that you do.
Yes.
It requires dedication and passion. Otherwise, and today there are so many demands on our time. There's family, there is, we have to look after. How did you personally manage that?
Yeah, I cannot sit at one place for a long time. That's my nature. And academically, I'm very happy. So I would like to spend as much time on academics rather than anything else. But I manage institutions and all that, regulatory committees, all that is okay. But my real pleasure lies in reading and trying to understand new concept and keeping pace in time and scale. See, now Government of India is in a different flavor now. As far as biology and these advanced technologies is concerned, a lot of support is there from the government now, unlike earlier. And a lot of money is also available for research.
Yeah, because money is a major factor.
Money is and also the supporting proactive government policies is very important, and the private industry also is ready to take on, able to compete with other developed nations. So I'm back by at least pushed back by about 10 years of this. 10 years ago, if this was available, it would have been a different issue. But now at least it's a lot of vibrant, a lot of churning is going on in scientific institutions. New concepts are emerging. The courage is now gathered to take challenges. So things are moving.
So future, you are optimistic about it?
Yes, 100% optimistic. There is no question about it. Whether the turbulences, speed breakers will be there, hiccups will be there. That doesn't matter. A nation of this size will just sit back and keep quiet. It won't do. That's for sure.
Fantastic. Thank you so much,
Thank you very
sir,
much
for taking this time, and it's been wonderful.
Thank you. Namaste.
Namaste.
Thank you for tuning into this episode of KanaCast. Please follow and subscribe to KanaCast on Spotify, YouTube, and Instagram. Until next time, woof woof.